advantage during evolution by protecting against tumors early in life, be it at the expense of limits to cell renewal later in life. So yeah, this is my disclosure statement. I started a company in 2006 to measure the telomere length in leukocytes, which is useful for detecting problems with telomere biology for clinicians looking after patients suspected of such problems. So to make a case, I guess, for the critical role of telomeres in tumor biology and aging, I need to briefly review milestones in evolution biology. This is being recorded. Let me just get rid of that. And I basically want to, I also want to review recent data on the accumulation of mutations in normal cells in relation to humans and in humans and in other species. I will review data on telomere length in leukocytes from my laboratory and repeat diagnostics next to exciting new findings regarding the structure of telomerase, which I just published using cryo-EM. And at the end, I will propose a new explanation for why women on average have longer telomeres than men, and how that could be related to the longer lifespan that women in general, or I guess on average, have. Okay, so before I start talking about telomeres, I should actually mention that go away. Before I start talking about telomere, I just want to mention that because I cannot refer to all the people being involved in this work. The main focus of my current research is not on telomere biology, but we did a lot of work, and I think it still stands the test of time. But I don't want to pretend that I did that by myself. It was actually the work by a lot of people indicated among others on this slide. In fact, the people on this slide were involved in the kind of work I will be presenting. Okay, so I'll start with kind of highlighting the milestones in evolution biology, and that always has to start, I guess, with Charles Darwin, who was, of course, is well known to all of you. He basically, let me try to get rid of this here as well. His famous book, The Origin of Species, so he proposed that all life is descended from a common ancestor, that the branching pattern of evolution results from natural selection, and that the struggle for existence has similar effect to the artificial selection involved in selective breeding. And all these things, of course, are now being accepted, I guess, in evolution biology. The second figure, which I think is underappreciated, but is also a towering figure, also in the 19th century, so a long time ago, is August Weismann. So he proposed that the distinction between the soma and the potentially immortal germline. And he also proposed that sexual reproduction with respect to generation of diversity of life. And finally, and this is, I think, prescient, and I'll get back to that at the very end, that senescence or aging results from both mechanical wear and a limit to cell divisions. And well, we'll get back to that. So in the 19th century, of course, people didn't know much about biology. Certainly, proteins were hardly or understood. There was no notion of DNA. But he basically proposed that there's a distinction between the germline, and he was referring to some material which are called germplasm, which was propagated and basically was propagated by the soma. And he basically proposed this barrier. It's a strict distinction between the immortal germ cell lineage produced by the gametes and disposable somatic cells, which was in contrast to Darwin's proposed Darwin didn't have idea about the separation between the germline and the soma. He just thought that animals reproduce, and how that actually worked, and how that would he didn't notice or he didn't mention this distinction between the two lineages. So in more precise terminology, hereditary information moves from only from germ cells to somatic cells. That is, somatic mutations are not inherited. He went to great lengths to give an indication of the kind of experiments that were done at the time, looking at mice where he would cut off the tails from breeding pairs, and the offspring would then have tails again. And just to make the point that such an abnormality was not inheritable, he repeated this kind of experiment 18 times, making the point that these somatic changes do not translate to heritability, and that there must be something separate, which he called germplasm, which is responsible for the transmission of genetic material, or I just say hereditary properties. Okay, so the third figure, don't worry, I'm going to leave it at 4 individuals, but it sets kind of the tone. This is also a key person in evolution biology, George Williams, and he's famous for proposing what he called antagonistic pleiotropy, which is that genes or alleles that control traits that are beneficial for an organism fitness early on in life, but detrimental to the organism fitness late in life, are expected to accumulate, because natural selection primarily works during reproduction. And if you have a proliferative or a selective advantage to reproduce, at the disadvantage later on, that these kind of genes and alleles will accumulate. And I think that's actually, we'll talk about telomeres in the context of this particular idea, and also actually talk about DNA repair in the context of this idea. So George Williams, he didn't like Weismann's ideas that mechanical wear can be involved in senescence or a cell division limit, and he basically said, well, it's ridiculous to think about mechanical wear as in machines or cars in terms of biological material. Although he published after DNA had been reported, he talks about genes and alleles, but not about DNA. Interestingly, he must have missed that. It was a few years after Watson and Crick. And he also mentioned that people had been looking for cell counting mechanisms for decades and never found it, so that's probably not true. And of course, we now know that there is a cell counting mechanism in the context of telomere biology, and we also know that mechanical wear or damage to DNA is a major factor in aging. So he was wrong on those counts, but he was very wise with respect to this antagonistic pleiotropy theory. Okay, the final figure I want to mention is actually Thomas Kirkwood. He's still alive, works and lives in Newcastle. And he basically combined Weismann's idea about this distinction between germline and soma at the time when he proposed this, together with Robin Holliday. DNA was well known and DNA damage was understood. And he basically proposed a disposable soma theory of aging. And the essence of this theory is that the amount of energy an organism allocates for somatic maintenance and repair is going to would not be more than needed to propagate, basically to reproduce. So this theory predicts that I don't know why I can remove this. Part of my screen is blocked by all these nice images of you. But anyway, the disposable soma theory predicts that unrepaired somatic damage accumulates throughout life, causing age and ultimately death. So that's I guess the current, still a very current theory on aging and why we age. Okay. So why doesn't it want to move on? So I think that's kind of the background on evolution biology that I want to present, and now I want to move on to new insight in aging in general, which comes from sequencing study, and particularly sequencing studies of single cells and normal cells or normal tissues. And I would say primarily work done by the Sanger Institute or people in the Sanger Institute in Cambridge, England, and particularly Peter Campbell and his colleagues, who I think made major contributions in terms of understanding how DNA is damaged during aging. So the summary here is this summarize what I'm going to tell you just by briefly highlighting these recent findings. Unlike what I was thinking, and I think a lot of people, mutations in DNA are not primarily reflecting errors during DNA replication. We'll get to that in a second. There's also this current data strongly support this disposable soma theory, the somatic cells accumulate mutations much more rapidly than cells of the germline. And the third interesting finding is that the number of stem cells in various tissues can be estimated from phylogenetic trees using the unique somatic mutations in individual cells. And finally, the number of mutations per somatic cell at the end of life in animals that are big or small, or that live or long or short, is very similar. So that's again support for this disposable soma theory. So one of these papers was published last year, where again, Peter Campbell and in this case, Abascal and colleagues were basically made libraries using, I guess, better ways to make libraries, looking at both strands, avoiding artifacts during library preparation and sequencing, and then basically measuring the number of mutations in individual molecules. So that's one way to look at mutational load, if you can do that. Use this method that they call nanoseq. Another way would be to take a primitive cell that can divide in vitro, and you can produce a lot of cells, and then you can sequence the progeny of that single cell and look at mutations in that progeny compared to the germline, and you can start counting. And people have done that, and you see mutations accumulate with time. But what's interesting that with this nanoseq, the number of mutations that you measure in granulocytes is very similar to what you measure in this progeny of a single cultured cell. And that's weird because there's a lot of cell divisions. Actually, they estimate more than 20 cell divisions. So that suggests that, again, cell division are not the primary cause of mutations. Well, interestingly, if now you feel that you can do this nanoseq, they've done it in different tissues, and you can see that all somatic tissues accumulate mutations at a rate of about 10 to 40 mutations per year. So that's kind of a clockwise accumulation of mutations, which is involved in aging. And importantly, cells that produce sperm, which you can measure, have a much lower frequency, an order of magnitude lower frequency of the accumulation of mutations. So what does that mean? This means that DNA repair in cells, testicular stem cells and the cells that produce sperm, must be better than in any somatic cell. And I think this is the first example of this antagonistic pleiotropy, where DNA repair, the energy devoted to DNA repair, was dialed down in somatic cells to allow for other properties that require energy, such as growth or other features that would increase reproductive success. So it's a sad notion that DNA repair in our cells is not as good as it can be, but this was a compromise that was made during evolution. So just to highlight how effective DNA repair is in the germline, you can look at these family studies where you look at parents and you can look at offspring and see how many differences there are at the genome level. And we now know that that's about 65 de novo mutations per generation. Most of those come from the dead. There's an increase with age, but it's still very low. If you talk about 65 mutations and people say reproduce when they're between 20 or 30, it's very low mutation rate in the germline, so remarkable. So the second thing about these normal studies of normal cells, and I mentioned already this possibility to grow cells and then look for mutations, that allows you to set up phylogenetic trees, because you can now do repeat sample and look for specific locations and come up with trees, this kind of barcoding of normal tissue, where, as I mentioned, the mutations accumulate kind of in a time-wise manner very constantly. But of course, the frequency of mutations will depend on when these mutations occurred. So you can use this actually to establish the number of stem cells. And I cannot go into how exactly this is done at this point, but the bottom line is that these kind of studies suggest that there's about 100,000 stem cells in an adult, and that these cells presumably divide very infrequently. So, highlighting some of the problems that we've always had to identify and study stem cells. They don't divide and they're very rare, and that complicates studies. But importantly, and I think this is going back to the first mention about mutation and cancer. So if we have about 10 mutations per year, and it's probably more like 20 in hematopoietic stem cells, and we have about 100,000 stem cells in a person, so then we can basically calculate that there's, of all these stem cells, there will be 3 million random mutations at 30 in the hematopoietic stem cells. Given that the genome is about 3 × 10⁹ base pairs, that means that there will be 1 mutation per 100 base pairs, or 100 mutations in a gene of 10,000 base pairs. So each gene will basically have been hit 200 times at age 60. And of course, now if you consider, not one person, but many, we see that it's inevitable that any gene will be hit at a location that will provide a growth advantage at age 60. Now, unfortunately, most mutations in genes, and we're talking only about single nucleotides variants, I guess, they will have no functional effect, or more rarely cause a loss of function. Because diploid genomes typically are, there's biallelic gene expression, so you typically can compensate loss of function by expression of the other allele. However, this is not true for rare gain of function mutations that provide cells with a proliferative advantage. And inevitably, over time, cells with such mutations will dominate stem cell population, even if there's very few stem cells, and they divide divide very little. And that's because the progeny of the cells will also have an advantage, and then the progeny of those cells again, so you get a kind of a logarithmic increase, and that will also increase the chance that these expanded cells will acquire secondary mutations. So that's a real risk for organisms that live longer. And in fact, that general notion is supported yet by another study from the Sanger, where they looked at gain of function mutations in normal esophagus. So they basically took biopsy of 2 millimeters square, and they, by targeted sequencing of 74 cancer genes, they then plotted the frequency of mutations that you can see here at the bottom in the normal esophagus. And you can see that, yeah, there's already evidence that in the 20-year-old of clones that are expanding, that becomes quite remarkable if you look at old individuals, and especially it gets worse if you look at smokers or drinkers. So there's a lot of mutations happening all the time, and it's it becomes important to prevent these things from forming tumors, and I think that's where telomeres come in. So again, you can, if you look at bronchial epithelium, and this is where they basically made organoids from bronchial epithelial cells. And yeah, you can see that this steady accumulation of mutations. So you have several thousand in a 30-year-old or an 80-year-old here. But if you're a smoker, yeah, you can add to the number of mutations. So there's both endogenous and exogenous causes of DNA damage, which cause the mutation. And in fact, the way we now think of these accumulation mutations is that there's primarily through metabolism, there's damage to thousands of lesions every day to DNA. Most of them are repaired, but every now and then things slip through at a rate of 10 to 40 mutations per cell per year in normal tissues. So that's an important thing to consider, that, and in order to increase that efficiency, probably possible, and we probably have the genes for it, but we decided not to spend that energy because we wanted to use that energy for other things. So that's the summary here. So disposable soma mutations accumulate at a 10-fold higher rate in somatic cells compared to cells of the germline. Mutations accumulate at a comparable rate in dividing and non-dividing somatic cells, 10 to 40 mutations per cell per year. I didn't highlight, but cells of the heart or cells of the brain accumulate mutations at the same rate as cells in the blood or the skin. So and of course, brain after 1 year of age, there's very little cell division. So that must really mean that it's this endogenous metabolism that is causing mutations and not so much cell turnover as we typically used to think. So somatic mutations are time dependent rather than cell division dependent. And of course, there's some limitations here. I only talked about single nucleotide changes. There's of course recombination events that can go wrong and stalled replication. There's other DNA damage, but this I think gives an overall decent and useful picture of the somatic landscape in cells. So you can increase the number of mutations by encountering specific DNA damage, as in smoking or drinking. So finally, in terms of sequencing studies, I want to mention this here. This study appeared just a few months ago that looked at somatic mutation rates in the lifespan of cross animals. So, and here what they looked at is widely different animals, with a variation of 30-fold in lifespan span and 40,000-fold in body mass. So from mice to whales, I guess. And then they look at the somatic mutation at the end of lifespan. And surprisingly, they find there's very little variation. So the way they did that, they basically they took colon crypts, assuming that these all are derived from the same colon stem cell. And then they measure mutations, and they do that at different ages of animals. And they basically count mutations that are unique to an individual crypt. And you can see, yeah, they steadily accumulate in all animals. And you can actually plot that. So humans have of course, pretty efficient DNA repair, because I mentioned only 10 to 40 mutations per cell per year. But in other animals, like in rat and mice, it's close to 800 mutations per year. And indeed, if you now measure the total number of mutations at the end of life, so several thousand typically, there's some variation, but it's much less than you would predict. And this points to an interesting conundrum, I guess. Why do mice develop tumors just as humans do, when there's so much more time and so many more cells that humans have to produce and sustain during a lifetime? So this has been puzzling biologists for a long time. So this relationship between body mass and cancer risk is puzzling. Some would predict that the risk of cancer should increase proportionally to the number of cells at risk of transformation. However, there appears to be no relation between body mass and cancer risk across species. This observation, which is called Peto's paradox, suggests that the evolution of larger body sizes require the evolution of stronger cancer suppressor mechanisms. And I would suggest that that's telomere biology. So I would say the answer to this question is Peto's paradox explained by differences in telomere biology between species. I would say yes. Low telomerase levels in somatic cells of large and long-lived animals triggers replicative senescence or apoptosis, which then results in a finite number of cell divisions, which is a tumor suppressor mechanism that is not present in small and short-lived animals such as rodents. This is, of course, a major problem in cancer biology. I mean, our favorite animal model to study cancer is the laboratory mouse. But if that's completely lacking this telomere tumor suppressor mechanism, then we're missing that whole aspect of biology, which as I'll indicate in a second, I think is very important for understanding human health and disease and cancer biology. So that then brings me after this introduction to telomeres and tumor biology and telomeres. So the bottom line, and I don't want to review this, I think you all understand or appreciate it, that cells of the germline express telomerase. They're kind of immortal. Telomeres don't really shorten. But in the somatic cells of humans anyway, telomerase is kind of limiting. And with each cell division, telomere gets shorter. And that's a good thing because it suppresses tumor growth. But it also is a bad thing because late in life, it suppresses the renewal of cells, and particularly cells of the immune system. So there are cells that can bypass this, and they typically do that by loss of p53, and then you get genetic rearrangement and typically upregulation of telomerase. But that again, that's a little bit outside what I want to talk about today. I do want to make the point that this function of telomeres, in terms of if the primary function, you could say, is to hide the end of a chromosome from the normal DNA damage response, and that's related to the length of repeats. So there's proteins specifically binding to telomere repeats. They form a structure which essentially hides the end from DNA damage. But this is a length-dependent function, so if there's insufficient repeats, there's insufficient proteins can be bound. And in fact, this end of a chromosome will look like a double-strand break, and it will activate DNA damage responses via ATM and downstream p53. So and presumably there's some repair, or there's some telomerase in many cells, including hematopoietic stem cells or lymphocytes, can deal with a few of these short ends. But if there's too many, basically then the cells will either die by apoptosis or become senescent. Now here are some of the dilemmas. So the telomere length is not a simple trait. There's 46 chromosomes in a normal cell, so there's 92 telomeres. And we can measure the average length pretty accurately, as I'll show you in a second, but that doesn't really inform about the shortest telomere. So we are assuming, and that's probably correct, that there's correlation between the average length and the probability that there are short telomeres, but that could vary between individuals. Now, telomere repeats are lost by different mechanisms, primarily through DNA replication, but there's other things like DNA damage by oxygen or replication forks that have a hard time going through R loops or other secondary structures like quadruplex structures, etc. Again, I won't go over this, but the bottom line is that telomeres do shorten despite that there might be some telomerase in somatic cells. So we spend a lot of time in my lab on developing these methods to measure telomere length. And the key here is that we use these PNA probes using in situ hybridization, whereby we take whole cells or chromosomes, denature the DNA, and then hybridize under conditions where the complementary strand cannot re-anneal. So we allow for what we call quantitative hybridization. And then you can measure the fluorescence on chromosomes, and you can now see, every end has a detectable amount of telomeres. It differs between chromosome ends, and the sister chromatids typically have a similar signal, which highlights the fact that you're hybridizing pretty close to in a quantitative manner, which is different than any other type of hybridization if you use RNA or DNA probes. Most of it, what I'm going to show you next, is based on flow fish, where we hybridize PNA probes to intact cells or cells which we denature in the lab, I guess, and then we hybridize PNA and we wash away the probe. So that's how this works. So we typically get blood samples. This is now done at Repeat Diagnostics, but they're no longer in my lab, I should mention. So if you want to work on this, you have to contact people at Repeat. But the bottom line is we get blood samples, viable blood samples. We don't want to use fixed cells because that complicates interpretation of this quantitative hybridization. We lyse the red cells, so we end up with lymphocytes and granulocytes. We add some cow thymocytes to that mixture as an internal control, and then we hybridize with the telomere probe for the PNA telomere probe, or as control we use no PNA, and we get background fluorescence versus specific fluorescence. You can see these cow thymocytes. They're useful as an internal control because they allow for calibration and variation between samples. They have always had the same telomere length, so we can calibrate whatever we see in lymphocytes, or in some cell types that we can measure, like CD20 lymphocytes. Most epitopes, I should mention, don't survive this denaturation procedure in 70% formamide and 85 degrees Celsius. So it's pretty harsh. Most proteins will fall apart. CD20 survives. And then what you can then see is that if you take the blood of a 5-year-old, telomere fluorescence is closer to what you see here for the control cells, the cow thymocyte, but in a 90-year-old, you see that actually the granulocytes, here measured by side scatter, are distinct from the majority of the lymphocytes, which in a 90-year-old will be typically memory cells, but you can still see some B cells or lymphocytes which are similar, which are probably primarily B cells here in this case. So if you plot that in hundreds of individuals over age, you can see that telomere length, actually what's not shown here very clearly is that the most rapid decline is early in life, the first few years when there's a lot of proliferation at the level of stem cells and lymphocytes, continues quite rapidly during adolescence, and then it continues to slow down or to decline with age in a very predictable manner. And the decline is more pronounced in lymphocytes than in granulocytes. In fact, we can measure some cells. I mentioned B cells, looks very similar to granulocytes. But what's interesting that the cells that show the most rapid decline, the most marked decline in telomere length are memory T cells and NK T cells. And naive T cells, in fact, also decline with age after puberty. And that's presumably because at that time, the thymus is lost and the naive T cells come from pre-existing naive T cells or other cell types. Okay, that is so far so good for telomere biology or telomere length biology. The critical component here is telomerase. Well, that's not a very simple enzyme, as I'll discuss in the next five minutes. There's very complex biogenesis, and there's very low expression levels, a few hundred copies of telomerase at most in a cell. So it's very difficult to measure. There's regulation at multiple levels, and there's differences in how this telomerase is regulated. In embryonic stem cells, the levels are limited by the levels of the telomerase RNA, whereas in most somatic cells, it's limited by the expression of the telomerase reverse transcriptase protein called hTERT, or encoded by hTERT. So Here we have these two major components for telomerase. It's the telomerase RNA gene and the H3 gene. The H3 gene is, there's a lot of regulation, we'll talk about it in a sec, but there's alternative splicing. Some of these splice variants are inactive. And of course needs to be translated into a protein. There's regulation at this level as well. But I would want to point out that third promoter mutations are the most common non-coding point mutations in over 50 tumor types. In fact, it's the most common non-coding mutation in cancer, period. Okay, so that just highlights, in my view, the importance of telomere biology in cancer biology. And for example, in cancer, bladder cancer patients, 60–80% of the patients have mutations in this mutation, which result in higher levels, I should say. Okay, so the other gene is this telomerase RNA. That's a complicated gene. It needs to be folded in a particular way, and the critical role in this folding is played by discarin, and I'll get back to that in a second. But after this primary RNA transcript is formed and folded, it needs to associate with the telomerase protein, which happens in the nucleolus, and then it goes into a further modification in candial body. So it's moving around, and again, very limiting number of molecules, and eventually then it has to find its way to the telomere. So complex biogenesis and complex multiple levels of regulation. So some insight has come from these new studies, which was just published 2 years ago, I guess now, on the structure, the cryo-EM structure of telomerase RNA. And what you can see here in blue is that there's basically two domains of this enzyme. You have this HAC ribonucleoprotein, which is stabilized by discarin, and you have this catalytic core where in green here, you have the reverse transcriptase. So if we focus in on this HAC ribonucleoprotein, it's interesting that you have the RNA here in blue, and you have two copies of discarin. And if you know, discarin is the first gene implicated in telomere biology disorders. If you look where these mutations map, they all map at the location where discarin interacts with the telomerase RNA. In fact, the telomerase RNA is sandwiched between these two of these discarin molecules. And apparently you need this, or presumably you need both the intact discarin to allow for this proper folding and thereby the stability of the telomerase RNA. And then this of course is a key finding, which I think everybody should really appreciate, that there's not many diseases where haploinsufficiency is autosomal dominant, but the telomerase, both third and third mutations can give rise to autosomal dominant disease, and you can measure this using the telomere length in the blood cells. So here we're looking at patients that are haploinsufficient for either TERT or TERK gene, and we're looking at siblings that are wild type. And you can see that there's a big difference between the kids in this case who have the mutations or haploinsufficient. So you can still readily detect telomerase in these cells, but it's about half what you would see in the siblings that have normal levels. So this enzyme level is very critical. There's still some enzyme, but if you reduce it by half, you run into problems in terms of telomere biology. This, in fact, is a patient that we studied in collaboration with Jeff Davis at Children's at the time, is that it shows typical spectrum. I mean, originally, this disease was called dyskeratosis congenita, and it's typically characterized by this triad of abnormal skin pigmentation, nil dystrophy, and leukoplakia. But that's not the main clinical problem of this patient. Typically is high risk of bone marrow failure, and MDS, AML, other cancers, pulmonary fibrosis, or other medical problems, even liver failure, for example. Frequency is rare because it's typically the genetic component of it. Interestingly, more common, and again, still maybe genetic, is pulmonary fibrosis, where that seems to be the same genetic mutation, but if you live past puberty, pulmonary fibrosis might be the major clinical feature. And again, these patients typically, pulmonary fibrosis, of course, is a major clinical problem, but they may also present with AML or MDS. And that frequency there's more patients with pulmonary fibrosis caused by telomerase mutations than this dyskeratosis. So, and then I would argue in the elderly, you get immune disorders, cardiovascular disease, and the frequency is very common. And that's just the spectrum of telomerase activity, where, if you're haploinsufficient, you have a little less. well, to some people, there will be some genetic variation in telomerase activity in the population. Yeah, just to mention here, so there's many genes now involved in this telomere biology disorders. Many of these genes play a role in this biogenesis that I mentioned. I mentioned dyscarrin was the first gene is X-linked. So there the females can be carriers and sons can be affected or unaffected depending on which X chromosome gene with the disease allele gets inherited. And I don't want to go again into this. So you can read up this. So for time. So the bottom line is this here. Telomere length at birth, and this is actual telomere length. So far I showed you in these plots telomere length calibrated with telomere restriction fragment analysis, which is kind of the gold standard. But if you look at the absolute length, absolute average length, at birth, telomeres in lymphocytes are longer than what you see in granulocytes. But by the age 20, that's reversed. So a lot of T cell production, I guess, is mainly T cells we're talking about here in the thymus, and there's going through more divisions. But at age 60, the lymphocytes are really getting, in a proportion of the population, very close to being too short to allow for further divisions, is what I would argue. And indeed, that's then what I call the telomere erosion, this disposable soma. So I would say that this disposable soma theory is correct, but for long-lived organisms, we need to include this telomere erosion component. And so there's genetic variation. This might be ongoing natural selection, if you want, or variation in population. So some people have very long telomeres, some people have very short telomeres. Very long telomeres might not be so great because then this tumor suppressor effect might be less. We mentioned that this senescence or telomere shortening is a good thing to prevent unlimited proliferation of these clonal populations before reproduction. If you're very short telomeres, then that might not be so great late in life, because then your immune cells maybe become limited in their ability to mount yet another immune response. And I should argue that there's papers making the point in large biobank studies in England where they looked at telomere length indirectly from sequencing studies and then look at risk of COVID, and they found that in fact telomere length is a predictor of COVID risk or serious, I guess, consequences of COVID independent of age. So that's an interesting finding. So if you have gain of function of mutations in telomerase, which gives you more telomerase, you have longer telomeres, you could say, well, that's great. Your immune cells won't have a problem later on. But unfortunately, it's actually not so great because you have a higher risk of clonal proliferation, because now the normal limit to clonal proliferation would not be implemented as it would be when your telomeres are average or short. On the other hand, if your telomeres are, and this would, for example, result from a loss of function mutation of TERT, then you would have increased risk of aplastic anemia or myelodysplastic syndrome, also clonal hematopoiesis, because a lot of normal stem cell clones would drop out, and that would then allow for these rare cells with maybe mutations in other genes, which give you longer telomeres, and you might also have increased. So that complicates the whole telomere field, which is already complicated by a lot of, I would say, inaccurate data based on PCR and a lot of commercial nonsense. But this is how I look at it, and I would certainly like to discuss that with you. So, summary of the second part, and then I'll switch to the final part, last 5 minutes. Clockwise, accumulation of mutations in somatic cells compromises the function of an increasing number of cells and contributes to aging. Alleles that suppress telomerase activity in somatic cells, such as H-TERT, promoter mutase allowed lifespan to increase by suppressing telomerase activity. I would think that's still ongoing evolution. I suspect there's a lot of variation in telomerase TERT, particularly TERT regulation, because you have this, on the one hand, benefit of longer life in immune cells, on the other hand, less tumor suppression, and that might be back and forth that's still playing out. And so while advantageous early in life, telomere erosion contributes to compromising cell renewal late in life, and both mutation and telomere erosion contribute to aging in long-lived animals. Okay, so now in the last 5 minutes, I just want to point out that our studies, and again, this is measuring average telomere length in leukocytes, or in this case, lymphocytes, If we look at cord blood, we see a difference. I mean, you appreciate there's a lot of variation. So some newborn girls who have shorter telomeres than some boys, and the other way around. But on average, there's a difference. And there's multiple independent studies which now make the same point. And then from our work, we see that this decline is very similar, in fact, between men and women, but the average length at any given age is shorter. So if you plot this, it's about 8 years in different. I don't particularly like this kind of calculation, but the point here is that maybe if lifespan is inhibited or is limited by your ability to mount immune responses, women might live a little longer. Of course, we don't really know what is the basis for this telomere length difference or the life expectancy difference between males and females. People postulate there's differences in cell division. Boys typically are somewhat heavier at birth than girls. Might be differences in sex hormones during development. But there could also be differences in telomerase expression during embryonic development. And why do I say that, and why do I think that? That's really based on studies that were published in 2016 by Rickard Sandberg and his colleagues at in Stockholm, where they looked at human embryos of different stages of development, so day 3 to day 7, and they isolated single cells, and they were either from female or male embryos. So you can see the numbers of cells that they analyzed, and they looked at they did single cell RNA-seq. And what they found is that if you plot gene expression, and you look, for example, of genes from the Y chromosomes, you don't see them in female cells. As you would predict, you see them in male cells. But interestingly, if you look at X chromosome genes, the expression level in male cells is definitely less than what you see in female cells. And we understand that X chromosome, there's typically during female development, there is inactivation of one of the X chromosomes, but apparently that doesn't happen right away. So during these early days, so day 3 to 7, there are still X chromosome or biallelic expressions from X chromosome genes. Autosomal genes, there's no difference between males and females. But this X chromosome, this delayed X chromosome inactivation is kind of interesting, because you can see day 4 here, there's almost twice the amount of gene expression from X chromosome genes compared to males. So what I did, I looked then in their supplementary data for DKC1, which, as I mentioned, is an important gene in telomere biology. And sure enough, the level of that DKC1 gene, which encodes for the discarin gene, is also much higher in female embryos than male embryos, and significantly higher. So I would propose that that's something interesting to look at. So we know that telomeres are longer in leukocytes from females compared to males, that this difference is already present at birth and is preserved throughout life. Delayed X chromosome inactivation in female embryos results in higher DKC expression in cells from female compared to male embryos. So there could be more telomerase RNA and higher telomerase levels in female embryos compared to males. And that longer telomeres in female embryos before onset of differentiation could result in higher reproductive potential of female cells at birth and throughout life. So that might result in women in delayed onset of immune failure and other senescent diseases. So there could, in fact, that's my theory here, which I just published a few months ago, that there might be, in fact, an embryonic origin for longer telomeres and longer lifespan in females. And I'd be very happy to discuss that and anything else I talked about in my talk with you. And I'll leave it at that and open the floor for discussion. So thank you very much for your attention.